friends, welcome back to my YouTube channel Astin Padasale. So in this video, I'm going to tell you top 200 important topics for your DBT JRF 2024 examination. So already I had discussed about the 70 days planner for this DBT JRF examination. So in that I had mentioned you, I'll be telling you 200 topics. If you cover 5 topics a day, then in 40 days, you can cover up almost half of the unit in this DBT JRF examination. So if you prepare the notes only for these 200 topics, means definitely for sure you can clear your DBT JRF examination in category number 1. So without any delay, let's get into the video. So first is RNA polymerase, like RNA pol 1, 2, 3 and their genes and which mRNA is transcribed by which RNA polymerase. So that tabulated column is available. Just study. Along with that, you need to know about the alpha amanitin sensitivity of this RNA polymerases. Okay. Next is DNA pol. Both in prokaryotes and eukaryotes, you need to know about the DNA polymerases. You need to know about template dependent, template independent DNA polymerases, like that. Next is enzymes that are used in rDNA technology. With special focus, you need to study about restriction endonuclease. Like there are 3 types of restriction endonuclease. You need to study about all the restriction endonuclease, 4 base cutter, and also there are few problems also there in restriction endonuclease. After that, you need to study about other enzymes that are employed for rDNA technology, like alkaline phosphatase, T4 polynucleotide kinase, like that. Next is types of RNA. mRNA, snRNA, snORNA, piRNA, tRNA, all that. In that, you need to study more about the tRNA. Next is types of DNA, A form of DNA, B form of DNA and Z form of DNA. Next, hybridoma technology for your monoclonal antibody production. You need to specifically focus on HAT medium. There will be many questions asked about B cell, myeloma cells, what is the role of hemopterin, like that. There will be many questions from hybridoma technology. Next is melting temperature of DNA. Next is histone. So histone in these days, you can expect in a single year, I think in the year 2019, there were 2 to 3 questions asked from histones. So study about histone with special focus you need to give on chromosomal organization. Okay. Next is virus that are causing disease in human, list of viruses that are causing cancer in human. Okay. So next is gene therapy. So complete gene therapy application, what kind of vectors used in gene therapy, suicidal gene therapy. Okay. So study more about gene therapy. Next is chromatography techniques, their principle, their application. You need to study about mobile phase, and stationary phase. Okay. Next is PCR, types of PCR and problems of PCR, and about the stack polymerases, list of things that are needed for running your PCR. So starting from a basic till this real-time PCR, you need to know. Next is sequencing technology, again Maxim Gilbert, till the spiro sequencing and next generation sequencing you need to study. Next is agrogel electrophoresis, starting from agarose, from which CV this agarose has been isolated, and then how plasmid DNA isolated from agrogel electrophoresis, and about gel percentage and size of your DNA. Okay. Next is SDS page technique, what is the role of SDS and about this procedure of SDS. Okay. Next is TI plasmid, the size of TI plasmid, virgins and their function. So simply you need to study a complete information about agrobacterium mediated gene transfer. Next is molecular marker, dominant marker, codominant marker, which marker is employed for DNA fingerprinting, like that you need to know, and RAPD, the gel format for your dominant marker and codominant marker. Next is marker assisted selection, is again highly important when it comes to DBT JRF examination. Next is with regarding to this carbohydrate biomolecule, like you need to know about disaccharide and their glycosidic linkage. Next, enzymes used in DNA replication. Okay. Next is amino acid. So complete information about amino acid, hydrophilic polar amino acid, which amino acid is consisting of sulfur. You need to know about the side chain, their isoelectric point of different amino acid in the side chain, and amino acid biosynthetic pathway. Like if you take glycine, the precursor molecule for glycine amino acid, like that you need to study about the biosynthetic pathway of your amino acid, and also you need to study about inborn errors of amino acid metabolism. So amino acid is very, very important. Just take notes only from Leninger for amino acids alone. Next Next is Ramachandran plot, again and highly important thing. You need to know about horizontal angle and all those things. And next, secondary metabolite, like there were different types of secondary metabolites. Study about all the secondary metabolite. You can refer textbook. Most commonly questions will be asked from textbook only. And also you need to study about economically important plant. So from you need to know about the secondary plant, the part of plant, and which secondary metabolite has been isolated from that particular plant. For example, Catherine roses, vincristine has been isolated. So in which group the vincristine, whether it is an terpene, whether it is an alkaloid, or whether it is an phenolic, and what is an application of vinblastine and vincristine? It is used as cancer therapy. So you need to know a complete tabulated form of the secondary metabolite. Next is vitamins, which vitamin, fat soluble vitamin, water soluble vitamin, metal containing vitamins, like that. And next is cell organelle, complete list of cell organelle and what kind of reactions are taking place in the organelle. And next is DNA barcoding technique. So DNA barcoding is again a more important question you can expect from DNA barcoding. Next is autoimmune diseases, cover about autoimmune diseases and which is in self antigen for that particular disease. Next is hypersensitivity reaction. There were 4 reactions are there. In QBbook you can find a tabulated column. Just read the tabulated column alone. You in these days in DBT BET exam also you can expect match the following type of questions. So don't compare CSIR question paper with DBT BET JRF exam question paper. So the difficulty level of DBT BET exam is below moderate level. It is easy to moderate level. So if you prepare for 2 months, at least 2 months you can clear in category number 1. Next is vaccine, a complete information about vaccine, DNA based vaccine, live vaccine, and which vaccine is given for this oral polio vaccine, and what kind of thing are being employed. Okay, so there were various vaccination programs are available, and you also need to know at which time point which vaccine has to be employed from baby, from birth till our puberty, what kind of vaccinations we need to take. So you need to know about the vaccine, the type of vaccine, at which stage of the life the particular vaccine has to be administered. And special focus you need to study about corona vaccine, like which cell line is employed for corona based vaccine, and whether Covishield is a recombinant vaccine or whether it is a live attenuated vaccines, like that you need to cover up. Next is regarding to plasmids and vectors. So vectors, in the sense you need to know about vectors that are used upon gene cloning. Vectors like you need to know about phage, BAC, YAC, which vector is used for human genome project. So you need to know about vectors, and also you need to study about viral vectors that are used for gene therapy, like which viral vector is employed for those cells that are rapidly dividing, which vector is employed for those cells that are not even dividing, they are terminally differentiated. So they were different. So study about viral vectors also. Next is animal cell line. So commercially important animal cell line you need to know. Antibody, so types of antibody, structure of antibody, pepsin digestion treatment, papain digestion treatment, and beta mercaptor treatment, the types of antibody like IgG, IgA, which antibody will be produced first, like that. Next is bioinformatics database like NCBI, DDBJ, EMBL. Next is BLAST program. Even though if you can't able to concentrate fully on the bioinformatics side, just concentrate on the database, primary database, secondary database, nucleic acid database, protein database. When it comes to protein, you need to know about protein structure database, protein sequence database. Okay. Next is cancer, complete information about cancer. So you need to study about genetic basis of each cancer, like proto-oncogene, tumor suppressor gene, and also you need to know about characteristics of cancer cell. Okay, so you need to know a complete information about the cancer cell. You also need to know about the therapies, current therapies employed for cancer, and diagnostic method that are currently employed for cancer. You need to know. Okay, types of cancer: leukemia, lymphoma, sarcoma, melanoma, all those. Next is transposon, like DNA transposon, RNA transposon, autonomous transposon, non-autonomous transposon, like that. Next is DNA, the complete information about nucleic acid, starting from measles and experiment until you need to know about entire information about the DNA, and pedigree analysis and the mode of inheritance. There are 4 different modes of inheritance, and sometimes they will be asking you cystic fibrosis is coming under which mode of inheritance. What kind of inheritance we can observe in cystic fibrosis? So you need to study about 4 inheritance pattern. So sometimes they will be giving you a family pedigree, and they'll be asking you to identify what kind of inheritance that particular pedigree is depicting, or sometimes direct questions will be asking. Okay, next, as I mentioned earlier, this is the pedigree, disease and their inheritance. Okay, whether it is an autosomal dominant pattern, or whether it is an autosomal recessive, or whether it is Y-linked, or whether it is X-linked, like that. Next is perions, their characteristics and their diseases, perion disease in human and animal. Next is microscopy. Starting from the principle till confocal microscopy, just read about the applications alone. Next is neurotransmitter, synaptic transmitter. So the syllabus itself, they had given 4 neurotransmitter, I think they had mentioned. Just study about the precursor molecule of those neurotransmitter alone. Next is DNA repair, types of DNA repair pathway, and what kind of diseases will be arising if that particular DNA repair is not functioning properly. So you need to know about the DNA repair diseases also, and then you need to know about immunological assay like ELISA, ELISpot assay, like that. Next is apoptosis. Apoptosis is again a highly important topic. Cancer, apoptosis, autophagy and all highly important. Okay, next is protein sorting signals. Okay, so protein sorting and membrane trafficking is very, very important when it comes to DBT BHR examination. So under the cell biology, you can expect these questions. So read about protein sorting. So in Albert cell book, they had given a clean information about this protein sorting. Okay, next is restriction enzyme, as I mentioned you earlier. Next is restriction enzyme, you can expect digestion related problems. So know about complete digestion, partial digestion. Next is circular dichroism spectroscopy, NMR, X-ray crystallography, cryo-electron microscopy. So these techniques are employed for determining a protein secondary structure and protein tertiary structure. So study about these 3 techniques compulsory. Next is triplet repeat diseases like Fanconi's anemia, Huntington disease, like that. You need to know the triplet, which triplet is repeated more in this particular Huntington disease, and whether that particular repeat is there in coding sequence or whether it is in non-coding sequence. So that thing you need to know. Next is IPR. I don't go in detail about this IPR, just read about IPR alone, types of IPR, patent, trademark, like that, example of those IPR. Next is flowering. So just read about flowering in control of flowering in plants. Again, refer Tice's book alone. No need to refer regarding to the flowering, control of flowering, just read Tice's book. Next is microbes and their industrial application, like which microbe is employed for commercial production of riboflavin, which microorganism is employed for commercial production of citric acid, like that questions you can expect. And also you can expect like which microorganism is employed for fermentation of your curd, fermentation of your dosa batter, like that you can expect. Next is centrifugation. So specifically you need to know separation of cellular organelle from by this centrifugation technique. Next is Alzheimer. So just read about the diseases like Alzheimer disease, Parkinson disease. Just read about these 2 diseases. So generally you need to know about diseases of nervous system. Neurological diseases is highly important for this CBT BHRF examination. Okay, next is Human Genome Project. So you can expect the questions from Human Genome Project. Next is chromosomal disorder. So while reading about chromosomal disorder, you also need to study about the cytological representation, like karyotype of all the chromosomal disorder you need to study. Next is familial cancer. So you need to know about the list of familial cancer. There are 5 diseases listed in DBT BET syllabus. Just try to find out. If you can't able to find out, just do tell me in the comment section. I can copy and paste you in the comment section. Next is cytoplasmic inheritance. So in genetics, there were different terms are there. Sex limited trait, sex influence trait, codominance, multiple allelism, pleiotropy. Okay, like that. You need to know about the example of each other genetical term. Next is chromosomal banding technique like G banding, R banding, C banding, and chromosomal aberrations like structural, numerical, euploidy, aneuploidy, like that. So both structural and numerical you need to concentrate more. Next is monoclonal antibody that are currently employed for the therapeutics of this cancer. Like which monoclonal antibody is commercially being given for breast cancer patient, you need to know about the generical name and the trade name. Next is immunostimulant transplant like xenograft, isograft, allograft, like that. Next is application of RNA technology. So from RNA technology, there were many things has been produced like your tissue plasminogen activator, erythropoietin. There were various drugs that was manufactured. But if you take humulin, humulin is also a product of RNA technology. So just read about the application of RNA technology products. Next is stem cells, potency of stem cells, induced pluripotent stem cells, totipotent stem cells, unipotent and their example, and multipotent stem cells like that. Next is diagnosis of this three diseases, TB, AIDS and malaria. How these three diseases are diagnosed, like Bidal test and other tests like for diagnosis of AIDS and malaria also you need to study. Next, sexually transmitted disease is important. Neurological disease, again it is important. Try to know about rabies also. And next is pyriaxal illness. Again, they had listed down the disease name in the syllabus itself. Just read about the disease name, note it down in your notebook, and just know about the causative agent for particular disease and whether there is any specific test has been employed for diagnosis or identifying means just write down them, and whether there is any commercial available drugs is there means you need to know about the drugs also. Next is monogenic disorder. Monogenic disorder, again in the syllabus itself they had given a list of monogenic disorder. Prepare notes only for that particular diseases alone. Next, inborn error of amino acid metabolism I had already mentioned. Next is microarray. Biofilm, which microorganism is involved in forming biofilm, what kind of staining technique is being employed for identifying this biofilm. Biofouling, with regarding to marine they are asking anti-fouling techniques like that. Next, biosensor, chromosomal manipulation in aquaculture. So biosensor is very important, friends. So try to read more about this glucometers also. Next is chromosomal manipulations in aquaculture alone. Just try to read. Next, immunostimulants. So if you can't able to cover up the entire syllabus, just take medical biotech and marine biotech in more serious form. So I dedicated at least 10 days for preparing notes for these two biotechnology related thing. Of course, from agriculture biotech also more number of questions will be asked. So just read more about, try to cover more number of notes for these three biotech: agriculture biotech, marine biotech and medical biotech. Next is bioremediation, bioaugmentation technique, marine pharmacology. So there were many anti-cancer drugs and other drugs has been taken from marine resources. So just read about the drugs or other compounds that are taken from marine flora and fauna. So marine pharmacology is an important topic. Next is biogas, biofertilizer. These things are also important. Next is major histocompatibility. So you need to cover about core concepts of classical life science systems like immunology, evolution. Of course, evolution there will be very less number of questions only will be coming. Just concentrate on immunology more. Okay. Next is antigen processing and presentation, fungal disease in human, protozoan diseases in human. You need to know about the vectors also, parasitic disease. Just try to read more about corona, malaria, and post-translational modification. Of course, post-transcriptional modification is also important. If you're already covered about replication, transcription, translation, molecular biology if you cover means just read the notes alone. If you didn't even touch your molecular biology means simply leave them, okay, because you have other topics for you to score well in this DBT bit JRF. Next is, as I mentioned earlier, you need to know about the virus that are causing cancer. Next is fluorescent activator cell sorting and cell cycle analysis by this FACS technique. Next is cell cycle, control of cell cycle like cell cycle checkpoint, and also you need to study about cell cycle with relation to cancer. Next is ABO blood groups, very important. Next is protein trafficking, Zika virus. So you need to know more about the Zika virus, coronavirus, Nipah virus, Ebola virus, and this flu. These five diseases are very important. After that, I had mentioned you about, before that, I had mentioned you about rabies. So try to cover. There are 10 diseases that are highly important, friends. So maybe in the next video, I'll be telling you the diseases that you need to take notes. Next is anti-cancer drugs. So when you are knowing about this anti-cancer drugs, you need to know drugs are chemical that are inhibiting microtubule. Drugs are chemical that are inhibiting or disturbing the function of actin. So simply you need to cover about cytoskeleton inhibiting drugs. Next is color. So sometimes they will be asking why Red Sea is red in color, why RK are purple in color, why grasses are green in color. So color based questions also you can expect. Next is eutrophication, enzyme inhibition. You need to know about the graph of enzyme inhibition and the example of each enzyme inhibition. Next is nude mice. Of course, models is very important. So cover more about this nude mice and skid mice and genetically knockout mice. Next is protein-protein interaction techniques and protein-DNA interaction technique. What kind of technique has been employed? Next is Yamanaka factor. Next is mutation, the types of mutation, all those things. So just read more about this mutation. Okay, mutation is again a very important thing. So don't leave this mutation simply. Next is RNases, RNase H, RNase I. RNases is very, very important. Next is with regarding to plant biology. So that is a tabulated column of the C2, C3, C4 and CAM cycle. Just read about the C2, C3, C4 CAM cycle and photosynthesis. If you are not well good in this botany side, you can simply skip out. Okay. These are the important topics from which you can expect questions. So I have informed you many topics in this video. So from this you can expect questions. 50% of questions will be asked from this topic they had mentioned over here. Because for not only for DBT bit, take for any exam, definitely one questions will be asked from this particular thing. And in DBT bit JRF examination, these topics are highly important. Next is nitrogen fixation, microorganism. You need to know about nitrogenase, enzyme, nitrate, reductase, nitrite, reductase, what kind of cofactors are involved. You need to know about nitrogenase enzyme in detail. Okay, what kind of cofactor is required, what is present, which metal is present in the active site of nitrogenase, like that. Next is genetically modified crops is very, very important. Just try to read about genetically modified mustard. Yes, recently government has approved for genetically engineered mustard. Next is immunology. Again, immunology, biology of T cells, biology of B cells. Next, try to read about immune cells like professional antigen presenting cells, non-professional antigen presenting cells. So immune cells and immune organelles are highly important. Next is cell signaling, broad subject cell signaling, but you can expect 2 questions from cell signaling. Just read about cell signaling. When it comes to cell signaling, you can go for Albert's cell book. Next is bacterial chemotaxis, quorum sensing. Next is cytoskeleton, lipid bilayer and protein sequencing. So when studying the cell biology, also you need to concentrate on protein transport, simple diffusion, facilitated diffusion, active transport. An example of those things: sodium, potassium pump, calcium pump, like that. Next is protein sequencing techniques like trypsin, which cuts in N-terminal N, and which chemical, which agent cuts in C-terminal N. You need to know about Edman's degradation method, like that. So protein sequencing is very, very important. Next is structure of protein: primary, secondary, tertiary and quaternary. So tertiary and quaternary structure is very, very important. Next is protein denaturation. What kind of chemical and what is the mode of action? Next is glycolysis, Krebs cycle, and also if you want you can study about pentose pathway, glyconeogenesis, gluconeogenesis, all those pathway. But glycolysis and Krebs cycle is very, very important. So my personal suggestion is that just try to cover up all the topic that you are familiar. If a topic is easy for you, just take a note, just give a read. Okay, so cover up the topic which you are familiar, which is easy for you. Just leave the toughest topic aside. So just believe the topics which you are going to study, from that only questions are going to be asked. Next is electron transport chain and inhibitors of electron transport chain, and how will you determine the concentration of DNA, RNA by UV spectrometry. Next is gene transfer method, both in plants and animals, you need to know. Next is reporter gene, simple selectable marker, screenable marker gene. Next is gene expression analysis, types of immunity: active immunity, passive immunity. And with regarding to vaccination, you need to know about active immunization, passive immunization, like that. Next is cytokines is again and very, very important. Complement system. So in immunology, you need to know about MHC, cytokine complement system. Next is plant hormone and telomere and telomerase enzyme. 2 things are very, very important. You need to know about human telomere, sequence of human telomere. Okay. Next is super antigen, herbicides and their target, and animal virology, like a list of DNA virus and RNA virus causing disease in poultry and livestock. Next is agriculturally important microbes. Next is protoplast fusion, tissue culture technique. Both the plant tissue culture technique and animal tissue culture technique, both are important. Give special focus for animal tissue culture technique. Next is types of fluid, like Newtonian fluid, non-Newtonian fluid, example of Newtonian fluid and non-Newtonian fluid. Next is antibiotics. You need to know from which organism the particular antibiotic was isolated and what is the mode of action of antibiotic. And also you need to study about translational inhibitor like tetracycline, furomycin, all those things. Next is antigenic determinants like isotope, idiotype, allotype, like that. Next is reducing sugar and non-reducing sugar. So don't leave this biomolecule concept. Study more about nucleic acid, protein and carbohydrate, vitamin. These four are highly important. Next is scientists and their discovery. Sometimes they'll be asking you who is the father of biotechnology, who is the father of fermentation, who coined the term CRISPR-Cas. Like that, you can expect more questions from the scientists and the discovery. And Nobel Prize winner is again and very, very important. Next is somatic hybridization technique and sex-linked inheritance, synthetic seeds, and about this ortholog and parallel. So I had mentioned you 160 topic. So remaining 40 topic I will be publishing you as a second video, because that 40 topics will be consisting of problem, because in DBT JRF examination 20% of the question paper consists of problem. It consists of subject related problem and aptitude. So the next 40 questions which I am going to upload as a part 2 will be consisting of important problems that you need to work out and important aptitude that you need to cover up. So thank you friends, thanks for watching this video.